Okay, this instructional video is for the Yokogawa LIFA spinning disc intravital spinning disc microscope. This microscope has additional functionality as well as confocality here in the facility. What this scope is really designed to do is live cell imaging in an incubated environment. So the microscope has the standard base that we see on the SB5 confocal and on our deconvolution microscopes. It's a standard Leica base. On the left port, there is a LIFA lifetime detector for frequency domain lifetime calculations, which will be covered in another video. The detection on this microscope is actually through the bottom port through a 512 by 512 back-thinned charge-coupled device by Hamamatsu. And before going to that charge-coupled device, there is a Yokogawa spinning disc confocal device. So, we're just going to cover startup now. The entire device is essentially turned on by two switches. We have a power bar located here for the device and another power bar on the floor that's located for the lasers. In addition, the lasers need to be turned on with a key lock safety interface. And for the 561 nanometer laser, the laser has to be turned on with the green button. And that should be seen by orange lights showing that the laser is on. So you need to manually check every one of these lasers to make sure that they've actually been turned on. This device has multiple laser lines. We have a 440 nanometer laser for cyan fluorescent protein, a 491 nanometer laser for green or yellow fluorescent proteins, a 561 nanometer laser for red fluorescent proteins, and a 650 nanometer laser for far red fluorescence. Okay, the other devices that need to be turned on the microscope are the mercury lamp for wide field observations for focusing, and a switch at the bottom in behind the thermal unit, the live cell unit, to turn on the heater for the incubator. And once the heater is turned on, we can select the set level, which is usually set at 37 degrees, and turn it on by pressing the green button. Normally this will take anywhere between 1 and 2 hours to properly warm up, so to plan the experiment ahead of time, you're going to have to come in and turn on the heater. What the heater is going to do is provide hot air through one tube into the chamber and recycle that air through the second tube and reheat it and put it back into the chamber. So during the warm-up phase, you want to make sure you have all the ports on the device closed off to ensure you aren't getting heat loss. Now for this video, we're just going to be looking at a fixed sample slide, which I've already placed onto the stage. The last step is that you're going to turn on the computer, and it's a standard Windows interface setup, and you're going to log in using the ADS server using your Mac ID. Okay. So once the computer has started up, what you'll see in the upper left-hand corner is the icon for the Velocity software package. We're going to double-click on that. And what will happen now is the Velocity program will start up and initiate its interfaces with the microscope. So for the sake of this experiment, we're going to create a new library by clicking on that icon. And I'm just going to enter today's date for this. And what you're going to see is a black box when you start up, and this represents the scanning area of 512 by 512 pixels. This is a back-thinned charge coupled device, which is a very sensitive amplified camera. You can increase the size of your viewing area by pressing on the zoom tool and clicking on the area. Each click doubles the size, and you can get that pretty large. You can go up to 400 or 800% of the surface area. If you want to back off on the zoom, control, click, you'll see a little minus sign come up in your icon, which could bring you right down to 100%. For typical imaging, I like to use 200%, which essentially fills the field of view. Okay. The next step is to then look across the various rows on the side here. This is the control panel for the device. It's not exactly intuitive, so we're going to have to run through it all. The most important panel is up here. This is actually showing us the optical configurations for the different channels of imaging. And you have the ability to do illumination of your sample by using lasers or by using the mercury lamp and filter sets. For the sake of finding your sample and focusing up on the cell of interest, we're going to use the wide field optics. So we're going to click on this icon, which will give us FTSE wide field. And what should happen is you'll see the wide field light turn on. You'll see this shutter open and close for your wide field sample, so you can manually control that so you don't bleach your sample before you go looking for it. And we'll now focus in on the sample. At the microscope, we need to change the port settings so that we're looking by eye. And in order to do that, we need to press this top left button here labeled BTP. BTP stands for bottom port. But if you click on it, you'll cycle through the ports on the scope, and you can see with one click, we now have 100% to the eyes. So if I look in, I can now bring the slide into focus. And the other button that's important is the Z fine focus. This will allow you to change between fine and coarse focus. So we turn the wheel away from us to focus up the objective. So the control of acquisition is really important on this panel on the side here, which has several different components. At the top row here, you'll see these colored icons, and they represent the different optical configurations, whether we're using wide field or confocal imaging. So we've switched over, we have it switched here to wide field. This is a FTSE, so if you hover the mouse pointer over the icon, it'll indicate what it is. There it's indicating DAPI wide field. Here it's indicating FTSE wide field. We click on that and we open the shutter, and we're now imaging using wide field fluorescence. At this point you can go over to the microscope and focus up your sample, but if you want to send the light to the camera, we press the BTP button for bottom port, and we now switch the light from the eyepiece down to the bottom port. All right, so this is the initial setting for the exposure is quite low, so we'll turn up the exposure. And we get our first cell image. And so right beneath the optical configurations are the camera controller. So this slider here is controlling the exposure on the camera. So right now it's indicated at 715 milliseconds. Right beneath that is the chip array, so you can control the binning that occurs on the camera. Right now it's set to bin 1. You're generally going to want to leave it at bin 1 because this is only a 512 by 512 pixel camera. If you increase the binning to bin 2, you're going to 256 by 256, and that's going to be pretty pixelated. And this control below that binning control is for the sensitivity of the camera. So you can actually adjust how sensitive the camera is. And with increased sensitivity, you are going to pick up more noise, so you should be aware of that. So you can balance between exposure and sensitivity to get your optimal sample image. Back at the controller here, we can now set the focus to fine. And using the top wheel, we can now change the Z to make sure we have our samples optimally in focus. All right. And then we can open and close the shutter to prevent sample bleaching, right? This mode is in wide field mode, even through the spinning disc, so you're going to see that room light does make an effect, and there is going to be some background on the room light. Okay, so essentially now we found our sample, we focused in on it. We have the option of looking at DAPI channels or red channels, either in a Texas red wavelengths or further CY7 wavelengths. But what we're going to do now is switch over to laser confocal settings. So we're going to press the GFP confocal button. And what you should see happening is once you press that button, you are no longer in bright field mode, you're in confocal mode. So the shutter is now down here, and that's the laser shutter. And this is the laser power levels, and we have 4 laser lines. And what's indicated here is which dichroic position is in place. So here it's dichroic number 2, which is appropriate for the green. And then we're going to have to adjust to see our image. All right, so now what we're going to do is we're going to switch into confocal imaging mode. So we were previously on wide field mode using the FTSE set of cubes. We're going to switch on to the GFP confocal icon. And what's going to happen is the shutter is going to open to allow the proper wavelength laser, the 491 laser, from going through on the system. We have the correct dichroic in place at position 2, defined as bright field and laser. And we have an indicator of the 40x objective and the emission filter, which in this case is emission filter position 2, which if we hover over that will indicate the GFP emission filter. And we get a very specific green fluorescent signal. Now we can adjust this in ways that, and to make it optimal. We may want to have, for example, much more sensitivity in the camera and use a shorter exposure time, and that'll help minimize photo bleaching. But what you'll find is with a spinning disc confocal optics, once you reduce that exposure time below 50 milliseconds, you're in fact going to start seeing some shimmering artifacts that occur in the imaging. And that's because the exposure rate is happening so quickly, you're in fact seeing the spinning of the micro lenses on the spinning disc. So optimally you want something that's a bit higher than 50 milliseconds, maybe as little as 100, or as much as 100 milliseconds. And that gets rid of that artifactual problem, and you can turn down the sensitivity on the camera. So once you're happy with those settings, you can now overwrite those settings into the computer by right-clicking on the icon and pressing overwrite. So from now on, these are the settings that will be saved for that channel, even if you bounce to other channels. This sample also has a red fluorophore. So if we switch over to the Texas red confocal channel, now what's happened is we've switched the dichroic to position, we left the dichroic at position 2, We've switched the laser now to the third laser, which is the 561 laser, and we've switched the emission filter down in the bottom to position 3, which is the Texas red emission filter. And you can see a nice red fluorescent color. Again, we'll adjust this to a little bit lower exposure, a bit more camera sensitivity. As with any camera, as you increase the sensitivity, you are going to increase the noise, but it's up to you to decide whether or not that's an acceptable level or not. Once we're happy with our level of exposure, once again we press right-click and we overwrite the settings. So now these settings have been completely saved for exposure, laser level, and camera sensitivity for this particular channel. So now we could bounce between the two channels, and those settings are completely saved. So there's red, and there's green. Right, so this is a live cell imaging camera, so what we really want to do is set up the experiment, which is for live cell imaging. First thing we're going to do is we're going to set up a simple 2-channel multi-channel imaging experiment over time. That is controlled on the upper panels up here now, where we have what's a red record button, which if we go onto that button and right-click, we'll get a pop-up menu, and where we want to go to is the acquisition setup for a particular experiment. Now in this case, there's already an acquisition in place here, but what I'm going to do is reestablish this. So basically what you can do is you can now set up a custom experiment. You can give it its own name. In this case, I named it GFP RFP 4-dimensional. You put in your username and you can put in a brief description so you understand exactly what this experimental design is about. And what we want to do is look at multiple light paths. So we'll click on this box here, change channels using light paths, and we'll add in our first channel. And our first channel we want is going to be GFP Confocal. The second channel we want is going to be the Texas Red 2 Confocal. And what we'll ask is that we do a time-lapse experiment. So we'll press Use, radio button, and we'll set the amount of time points that we want for the experiment. So this could be set in various ways. We could set it for maximum speed, which means we'll acquire between the two channels as fast as possible due to the exposure of the different channels. So you may want to do this in live imaging for a very fast event. You can set it to the number of time points per minute, or the number of time points per hour, or the number of time points per second, or the number of minutes per time point, for example. So I think what we're going to do is we're going to set this to time points per minute, and we're going to pick 6 time points per minute. So every minute we are going to take 6 frames of data. And we're going to not change the focus for this experiment. The focus is set at 1 level, and we're going to say OK. And that's what the experiment is. So now when we go back to our image, and you can see in the entire time I've done this explanation, and we watch these cells, we're in fact getting very little photo bleaching, even though we're doing a continuous illumination onto the field. And that's one of the major advantages of using a spinning disc confocal microscope. So we'll start the experiment now by pressing the record button. And what this will do is start the acquisition until we decide to stop it, and which means we're going to have 6 acquisition points per every minute. So every 12 seconds, and you can see that's counting down up here, we're going to capture an image. And this will continue to occur until we press the stop button. And that's a simple multi-channel acquisition. Now we can actually look at this by double-clicking on the library, and we can see the image of interest. We'll zoom in on it. We're actually looking at the 2 channels overlaid here, and we can adjust this as we're looking at it. So we can turn off the GFP, for example, or turn it on, or turn off the Texas Red, or turn it on. We can adjust the brightness in each one of the channels. That'll allow us to, optimal imaging quality. And we can go through the time course. Now in this case we happen to be looking at fixed cells, so there's not going to be much to look at, but as we do press play, we can in fact cycle through the different times of the experiment and actually watch the experiment as it occurred in 2 channels simultaneously, or any one channel at any one. Another potential way to do this experiment is, instead of the investigator having to sit at the microscope and decide when to stop the experiment, is you can do the experiment over a set time course. So again, right-clicking on the record button and going to acquisition setup, we can then set the time lapse button for a certain number of time points. So for example, we chose 6 time points per minute, and we can decide that we want to do this for a total of, in this case, 2 minutes. But you could set it for minutes to hours, of wherever you want. So for example, if you're going to do time course observation of drug treatments, you can do it for a set time of 1 hour or 2 hours and program it into the program. So if we do this now under this acquisition setup and press OK, what'll happen is, if we start the experiment using the record button, it'll only take place over a period of exactly 2 minutes. All right, so what we've seen now is in the process of this 2-minute time course experiment that we've programmed in, it's going to stop its acquisition when the time stops at 2 minutes. All right, and if we now look at this experiment by looking in the library, we can actually see on the 2 channels more acquisition times as we go through time down here. Now, if you're doing a very long experiment on the order of hours, you can actually watch what had happened in the past during your time course experiment while acquisition is occurring on the microscope. So you can essentially do a preview of your experiment before it's ended by clicking on this panel, and the computer and the microscope will still continue to do its image acquisition. Okay, so we're going to stop this, close this panel, and go back to our image acquisition panel. And now what we're going to do is engineer in what's called a 4-dimensional experiment or a multidimensional experiment. So what we've shown you is how to do 2 channels, and you can add more channels if you want, at one set time point. And what we're going to do is now match this to a Z stack in 3-dimensional space at those time points. So the way to do that is, we're going to pick one channel, and for sake of convenience we'll pick our green channel, and now we're going to focus in on this controller here, which is for the Z focus controller on the Leica stage. By left-clicking, just one click onto this controller, once we like our focal plane here, we're going to set this focal plane to 0. And this is now essentially the midpoint of focus. Then we're going to manually, using the focus controller, back off that focus to the top of the cell, click on it again, and we're going to say set top. And we're going to click through the other direction, and we're going to say click on it and tell it to set bottom. And then you can click on it again, and instead of saying set, you can say actually move to those positions. So move it back to 0, and it'll go right to your proper focus plane. You can check that your top is correct by saying move to top, and you can see we're at the top of the image. Click on it again, move to bottom, and it's going to focus to the bottom of the image. So for now we'll just set it, move it back to 0 position. So we now have defined the parameters of the focus between the top and the bottom of our Z dimension. I'm going to close the shutter so we're not bleaching our sample. And we're going to go to our record button again, and again right click on that and go to acquisition setup. So here's the old acquisition setup where we had the experiment where we were doing 6 time points per minute for a total of 2 minutes. And now what we're going to do is add in the option to change the focus. And the default is none. And we're going to pull this drop down menu and tell it to change the focus using the Leica focus drive. And there's 2 options to do this with now. It knows the range from what we set in here. We can say either capture with a set number of slices within that range, or we can choose the option of choosing the Z spacing of those slices, right? For the sake of convenience, I'm just going to say capture with 12 slices. Leave the scan direction as up, which is recommended. There is no autofocus, don't bother with that. And then what you really want to be careful for now is the managing of your shutters, right? And there's multiple options here that are not necessarily intuitive. So in this drop-down menu, what you'll see is maximum sample protection, balanced sample protection, maximum speed, or don't manage at all. For these types of experiments, you really want to set them for maximum sample protection. And what that'll mean is when one laser in between image captures at the various stages of the experiment, the shutter is turned off and no light is allowed to go into the cells. If you absolutely want to capture at the absolute fastest speed possible, due to the biology of your experiment, you want to set this to maximum speed, which essentially will not use shutters and simply switch between laser wavelengths. So for this experiment, we're going to click maximum sample protection. And then we're going to say OK. And once again, with every experimental difference, you can save it under a different profile and restore those profiles in the future if you want to just reproduce the same experiment again. So now when we press on the record button, the experiment is we're going to do 6 time points per minute for a total of 2 minutes. We're going to choose 2 channels at each time point, and we're going to go through the Z stack at those time points. I'm just going to go back into that to confirm all of this, and notice that there's a way that we can also grab the Z sections. So there's 2 options, Z first and then the channels, or channels first and then Z. So basically it depends on how fast your system is moving. Z first and then the channels. What'll happen is it'll capture a Z stack in green, and it'll go back, change the laser wavelength, and capture the Z stack in red. If you have a very fast event going on, such as vesicular trafficking, you may want to do channels first and then Z. So what'll happen is at every position in the Z, it'll switch between green and red as it's going down in the Z plane. For the sake of convenience in this experiment, we'll just choose Z first and then channels. So at the bottom of all of this, as you make these changes, what you get is a very nice summary explaining exactly what the experiment is. So we can read this here. Capture for 2 minutes. Acquire at a fixed rate of 6 time points per minute. Capture 2 channels by changing the light paths and 12 Z slices. It even tells you what the micron step size is. Using the Leica focus drive upwards. Capture multiple Z planes for each channel. Shutters will be managed for maximum sample protection. So you should read this because it'll tell you exactly what's going on, and if it doesn't match with what you want, then you're going to have to change the parameters to get what you do want. Again, we're going to press OK, and we're going to start the experiment. So what's happening now is you're going to see the focus changing in the Z, then it'll switch over to the red, then the shutters will turn off. And what you can see for this type of experiment, because it takes a certain amount of time to go through that Z stack, it's essentially become continuous because it takes that amount of time to essentially do 6 time points per minute. And so what's going to happen is now we're going to have a 4-dimensional data set that actually has multi-dimensional channels, so really 5 dimensions, because we're looking at X, Y, Z in terms of 3-dimensional space, and 2 channels of color for each one of those time points. So we're going to just let this experiment continue to capture, and then we'll visualize it at the end. All right, so now that the experiment's been completed over 2 minutes in the 4-dimensional experiment, this is how we can look at the data. If we double-click now on the library, what we have is our 2 channels of fluorescence, so we can optionally look at both of them or just any one of them. Along the bottom, we have our time course during the experiment, so we can move to any point in time. And using the wheel on the mouse, we can scroll through the Z dimension and look at the different focal plane. And this is one way to look at 4-dimensional data. We can then export this data set into various software that will actually allow you to visualize it as a 4-dimensional data set. But this is a way to look at it in the scope. And once again, as with previous experiments, you can actually look at this while acquisition is continuing to take place during the experiment at any time. Okay, so for completion of experiments, it's a pretty simple procedure. All you have to do is shut down velocity, shut off the computer, and reverse the order of shutting down everything at the level of the microscopes and the 2 buttons for the 2 power bars. And that's pretty much completion of how to do imaging on the spinning disk confocal system here in the MBF.